Okay, so in biotopic 7, we learn about DNA replication, what is it, translation, transcription, and at least in 7.1, what is very important is the DNA. And if someone asks, how do you know that the DNA is the genetic material of living organisms, we can talk about this Hershey and Chase experiment. So in the past, the controversy was whether the DNA was the genetic material or whether it was the protein. So Hershey and Chase designed the experiment to prove that DNA is the genetic material, and what they used is the bacteriophage, and this is a virus. So since it's a virus, it has either DNA or RNA, and this one had DNA here. So let's say this is the DNA, and this is protein. The outer layer is the protein. Okay, so now we'll just divide this into two parts. They conducted two different experiments, or pretty much the same thing, but a bit different. So in here, they labeled the DNA with phosphorus 32, and this is a radioactive isotope. So they labeled it with phosphorus 32. So they labeled the DNA with phosphorus 32. Remember that the DNA has a structure like this, right? And then a nitrogenous base here, and so on, and the other way here. So it has phosphorus and nitrogen, and they chose to use phosphorus here. And this is radioactive because intentionally, because then we can look at where this DNA is through radioactivity. And this side we label the protein with sulfur 35. This is also a radioactive isotope. And if you consider the quaternary structure of a protein, probably you learned it in topic 2. So these are just amino acids. So the polypeptide chain, and there are disulfide bridges here that hold on to these proteins together that give them this kind of weird structure. So therefore, we can say that in protein there are sulfur. And knowing this, Hershey and Chase used sulfur 35, which is radioactive, so that they can track the radioactivity to see where this sulfur is. So basically where the protein is, DNA. Okay, and then now that they labeled it, let's also change it up a bit so that we know what we're talking about. Here. So we'll label the protein with green, like this, and we'll label the DNA with red. No particular reason. Okay, and now we know that bacteriophage infects bacteria. That's why they're called bacteriophage. So they infect infect bacteria and so that they can reproduce and make more of themselves, because they're a virus, they cannot reproduce by themselves and they need some kind of host to make more of them. So this means that they pass their genetic material to the bacteria, and this bacteria is E. coli. Not really relevant, but it's usually most experiments are conducted with E. coli because they're not too dangerous. We can find them pretty easily. Yeah, so like that. Or at least we didn't know this, but now that we do know that the DNA is the genetic material, we can sort of draw it like this, but let's just leave this aside for now. Let's just say that infected also infected. Okay, and now these bacteria are put into like a test tube. And then we centrifuge them. Centrifugation is like spinning really fast. You probably also saw it before in your school lab. It's like here, and then you can put little things here, and then this spins like really fast. And what happens is that heavy materials are here and the lighter materials are on the top, right? So since the bacteria is much bigger than the virus, and so it is much heavier than the virus. So if we compare that, we can expect that the virus is here and the bacteria is here on the bottom. And when we tracked the radioactivity that we used to label these, the protein and the DNA, when we tracked them down, we saw that the labeled phosphorus 32 was actually seen in the bottom half, and the labeled protein was seen in the upper half, or not necessarily half, but upper, and that the phosphorus was seen in the lower. But we know the bacteria is heavy, so it is bottom or lower. And that this has genetic material of virus. And we labeled them with phosphorus and sulfur. But since sulfur is on the top, phosphorus is on the bottom, we can know that bacteria is on the bottom and phosphorus is also at the bottom, so the genetic material has to do with what was labeled by phosphorus, which is DNA. So that's pretty much how we know that DNA is the genetic material. And that was the Hershey and Chase experiment.